Welcome everyone. We just wait for more people to arrive and then we will start. Okay, so I think we can start. So welcome everyone again. Thank you for coming during the holiday break, it appears. So today it is my great pleasure to welcome our speaker, Adam Cripps, who came here all the way from Oxford. Well, luckily, we don't need to travel now.
Yeah.
So Adam finished his PhD in Imperial College London in 2013, and then he did a postdoc in the University of Oxford working on the epigenetics of T cells. After that, he was successful to obtain a highly competitive 3-year MRC fellowship to establish his research program in computational biology, also in Oxford. And this was very successful. And after that, he was awarded a second MRC fellowship grant to move more into cancer. And today, Adam will be talking about both of these directions, I guess. Well, now the floor is to Adam.
Oh, thank you. So let's start that. Can you see that, or have I clicked the wrong button again? Wait, actually, I think before I clicked the share desktop because it didn't work, did it? Perfect. Cool. So thank you. What I wanted, I expected today to give a talk on purely focusing on the single cell sequencing research that I'm doing related to multiple myeloma, but actually I thought I'm a little bit bored of just talking about single cell, because really for me, single cell is just one tool in many tools that I use to ultimately understand the molecular mechanisms underpinning healthy and disease. And so I'm hopefully going to try and interweave 2, 3, maybe 4 different stories into one presentation that will hopefully flow, because I wanted to give you a bit of background on why we study single cell, because sometimes that's not always apparent when you go to single cell sequencing talks. So as Vlad said, I did my PhD in the late Professor Fionnuala Brennan's group. I was working on molecular immunology in the wet lab, and then undertook my postdoctoral position with Mark and Udo in the Botany Research Centre in Oxford. Again, wet lab research working on epigenetics of T cells, but it was during this time that I actually became really frustrated with science, because I was generating some fantastic data, high dimensional, next generation sequencing data. And unfortunately, I just didn't have any of the skills that were required to analyze any of the resulting data. And this ultimately acted as a catalyst for me to successfully, and I was incredibly lucky to have applied for an MRC Career Development Fellowship in computational biology within Chris Ponding and David Sims' group in the CGAT program. So it's the Computational Genomics and Training Center within the WIM. Here I learned lots of new statistical approaches, software development, and then generating workflows to aid the analysis of next generation sequencing data. Following this, I then moved to the botnet to set up my own group. And again, incredibly lucky during the unfortunate climate we have at the moment, that I was successful in January at receiving another MRC Career Development Fellowship, this time to move the direction of my research away from autoimmunity, more towards cancer. And this is what I'll give the talk on today, specifically myeloma. So because I'm a computational biologist now, I'm in a really great privileged position to be able to work across multiple different projects. And so I work across the Oxford Bayer Consortium on Women's Health. So I get to work with these great people down here. In addition, I also work as a team member within the Celgene Epigenetics Consortium, specifically on myeloma, and I'm a junior investigator on the Leduc atherosclerosis project with Uda Opperman. In addition to this, I'm the computational lead on the Tendon Seed Network, which is it forms part of the Human Cell Atlas project, and this is led by Sarah Snelling here. I also, as I said, moving my research more towards cancer. So I work with the Bone Cancer Research Trust with Lucia Cotone on chordoma, and also medulloblastoma with Tuba in Turkey. But again, as I said, I'm now focusing most of my research towards multiple myeloma and single cell sequencing technology development. So the tagline for my group is ultimately my group is trying to understand human health and disease biology from a functional genomics standpoint. So using lots of next generation sequencing approaches, but also some very basic biochemical and biological approaches, as well as cell culture techniques. And the talk I want to give today is I want to talk more about the compound library approach we are taking to identify novel epigenetic targets. I'll give an overview of two of these, one which is an epigenetic target, GSKJ4, and one which isn't, but they're linked together in terms of their mechanism. Then I'll talk about single cell sequencing and how we're using single cell sequencing in the myeloma context. And then the last 10 minutes, I'll give an overview of some of the new technology I've been working on to develop long-read single cell sequencing, again, to help understand multiple myeloma. So you can think of this as the lab's wet lab pipeline. So we tend to work at the beginning of the pipeline by identifying targets that we want to work on through either CRISPR shRNA screens or chemical screens. We then identify targets, typically in the epigenetic field, and then we leverage this with the latest molecular diagnostic techniques, that's single cell sequencing, next generation sequencing approaches, CyTOF, etc., and basic cell culture techniques. To integrate all of this data together, we leverage this with high performance computation. So I spend a lot of my time developing workflows in the computational space to analyze, streamline the analysis of this data, then integrate it with not just different modalities of data, but also with different public data sets. And then I have very good clinical collaborations, a lot of them within Oxford, so it's very easy to get a good clinical perspective when you have clinical collaborators that work very closely in proximity with you. And so this in myeloma field is Karthik Ramazami and Sarah Gooding. So I want to talk about the chemical tools that we use within the lab. So majority of these chemical tools are developed within the Structural Genomics Consortium, which is a large public-private partnership, which includes 10 academic centers and 9 different pharma companies. And I suppose the bread and butter of the SGC is they crystallize proteins, typically at the diamond light source, and then determine the structure. And then they have very clever chemists who then come along and then develop small molecule inhibitors to inhibit the protein. And typically the SGC is focused a lot of their effort on the epigenetic machinery. This is the readers, the writers, and the erasers of the histone code. And this is, all these compounds are curated with Uda Opperman. He's a PI at the SGC. So we actually have compounds that target a wide range of different processes. Again, I've mentioned epigenetics, which has been the big focus for the SGC, but they also have kinases, PROTACs, compounds targeting metabolism, and ubiquitin proteostasis. And these compounds are constantly updated and expanded, and this is Jim Dunford's in charge of curating all of this. So when we have all of these compounds, the first thing that we usually start with is a very simple screening approach. In this context here, because of my background in autoimmunity, I was looking at Th17 cells, which were a pro-inflammatory immune cell that are very important for diseases such as rheumatoid arthritis and ankylosing spondylitis. And we identified GSKJ4 as a hit for reducing IL-17 expression within this system. And what GSKJ4 is it's a molecule which inhibits GMJD3. And so this GMJD3 enzyme, its job is usually responsible for removing methylation marks at the H3K27Me3. And this mark usually represses gene expression. So if you inhibit this enzyme, you should see an accumulation of K27Me3 across the whole genome. And that's actually confirmed with chromatin immunoprecipitation, just in this figure. The blue shows you the GSKJ4 and the red, the DMSO. What we try and do when we develop inhibitors is the SGC develops an inhibitors, they always try to have a stereoisomer control. So you can see GSKJ4 here, and it has a stereoisomer, which is a chemotype, which is very similar chemotype called GSKJ5. And the only difference between these two is the position of this nitrogen in this ring. And that's enough to inactivate the enzyme by reduce the potency of the compound by 300 times. So we usually add this in as a good negative control within our system. So it has anti-inflammatory properties, but I've also been working on chordoma with Lucia. We published a paper last year where we found GSKJ4 also inhibits chordoma, so chordoma cancer. So chordomas are cancers which develop from the base of the skull all the way to the base of the spine. They can develop at any point along the spinal cord. Chordoma is a really horrible disease, cancer to get, because it's so close to the spinal cord that the only real treatment is surgery or radiotherapy. And because it's proximity to the spinal cord, typically any surgery usually renders people in wheelchairs. In addition to this, it's also a very, very slow growing cancer. So unfortunately, chemotherapies just don't have any effect. However, what we found was that GSKJ4 was able to silence a gene called brachyury, or TBXT. And this TBXT is the gene which is involved in driving chordoma progression. And we found that when you add GSKJ4, you get a global increase, or you get an increase in H3K27Me3 around TBXT. And this silencing allows of TBXT ultimately kills the cancer, so the cell's no longer to maintain its proliferation. So as I mentioned, most of my research now is focused on multiple myeloma. Multiple myeloma is a plasma cell malignancy, so it's a hematological disease, and it arises through the proliferation of abnormal plasma cells within the bone marrow. Patients typically have a survival of around about 5 to 7 years. And the disease follows very nicely, typically a Darwinian evolution principles. So what ends up happening is that the cells proliferate so quickly that they end up developing drug resistance very easily. So there's a number of really good treatments that are available now. But when you then treat a patient, they go into remission. And then after a period of time, then they come out of remission and then they get full-blown myeloma again. And you go through a series of rounds of treatment and relapses because drug resistance is a significant problem. And so that's my work, is trying to understand from the functional genomics perspective, particularly the clones and the mechanisms that are involved in this drug resistance. So again, going back to the epigenetic screens, we typically start with a very simple readout. And this is, we've screened all of the 900 and odd different compounds across several different myeloma cell lines. And I'm not showing you the drug resistant cell types today, but we've also done those. In the myeloma models, we show killing response. So these are just the compounds which we've identified as being hits for killing myeloma cells. And again, we see very nicely that GSKJ4 is a top hit and it's able to kill myeloma. Typically, the first thing we do is we go and do very simple assays. So this is just making sure that the compound actually is killing the cells and it's not due to some, halting proliferation or typically a halting proliferation or a change in metabolism. So here you can see using flow cytometry with a Nexin-5 and PI, which is indicative of cell death or cells dying. You can see with GSKJ4, you have an increase in the double positive cells compared to DMSO. One of the things that's really great about this is, the assays allow you just to count the cells, which is really useful for when you're looking at killing. You can look at it in lots of different complicated ways, but really, how many cells are in the culture well? And you can see that with increasing amounts of GSKJ4, this leads to increasing numbers of cells dying within co-culture assays with immune cells. So to really understand the basis of what is happening when you add GSKJ4 to myeloma cells, we typically start with a bulk RNA-seq approach just within our cells to see what is happening. And what we can quite clearly see is even at very early time points, and I'm talking 1 hour to 2 hours, you see this huge increase in a metallothionein response and a increase in ATF4 family transcription factors or downstream response to ATF4 transcription factor. The reason why this is interesting is because ATF4 is typically a stress response. It's activated in response to amino acid starvation. And there's a cascade of signaling that occurs, and it's usually kicked off by phosphorylation of EIF2 alpha. And you can see here with GSKJ4, we get an increase in phosphorylation. And then we have an increase in the protein expression of the typical ATF4 family of transcription factors, that's ATF4 and CHOP. And this then initiates an amino acid response, which tries to recover. The cells try to recover from the amino acid stress response, but in the case of myeloma, they obviously can't. So we can see this quite clearly. We see an increase in amino acid metabolism in response to GSKJ4. In addition, we also see an upregulation of amino acid transporters. So the cells trying to respond to the activation of ATF4 by upregulating these amino acid transporters. This leads to a huge change in the metabolic function, which I'm not going to go into today, but you can see clear changes in the way the TCA cycle is being utilized. And then it leads to the induction of cell death in myeloma. Interestingly, we don't see the response in the immune cells. The immune cells can tolerate GSKJ4 very happily. Even though we do see an ATF4 response, the cells are able to recover from it. So just the conclusions on GSKJ4 before moving forward. So I've shown hopefully that the compound library screening allows us to identify potential therapeutic targets. In the case of GSKJ4, the therapeutic target is potentially the Jumanji histone demethylases, which have both anti-inflammatory and also anti-cancer effects. But what I want to stress is that even though it's the same compound and the same target, the effects that we're seeing within different model systems are very different. For instance, we see the induction of amino acid stress response very clearly within the myeloma cells, but we don't really see that within the inflammatory cells or in the chordoma, where it seems to be a direct consequence of, in the chordoma at least, the downregulation of H3K27Me3 across the brachyury region. So moving on from GSKJ4, one of the other hits which we found really interesting and wanted to pursue was halofuginone. You can see here, it's one of the top hits. It's MAS1392. The reason why this is interesting for us was because halofuginone inhibits prolyl tRNA synthetase. And this is a compound which is actually in clinical trials for antifibrotic and anti-inflammatory applications. And you can see on the, Katrine in the lab crystallized the PRS complex, the PRS and showed that the inhibitor sits very nicely within the tRNA proline site. So the reason why this is interesting for us is because we saw this paper in Th17s, which showed that you can actually affect the differentiation of Th17s by inducing and activating an amino acid starvation response, which looked so much similar to the response that we were seeing within GSKJ4. Unfortunately, GSKJ4 is never going to be a therapy. The compound is just not suitable for in vivo applications, but halofuginone potentially is. So just as a slight segue, just to show you that halofuginone again has different effects depending upon the different system you have. I want to talk quickly just about a collaboration that we've had with Philip Gortz within the UCSD. So himself and Jeffrey Esco published quite early on at the end of last year, that SARS-CoV-2 infection is actually highly dependent upon cellular heparan sulfate. So when you have heparan sulfate, it aids the attachment of SARS-CoV-2 to the ACE2 receptor and then aids the viral uptake. I've been working with Philip on the atherosclerosis project for a number of years, and they have our epigenetic inhibitor libraries, and they just profiled some of these compounds on the ability of SARS-CoV-2 to be taken up by Hep3B cells. And they found that halofuginone is, again, a top hit at downregulating this heparan sulfate response. Interestingly, it activates the EIF2 alpha stress response, but unlike in our system, halofuginone doesn't, the stress response doesn't actually play a role in inhibiting the either the SARS-CoV-2 entry or actually the SARS-CoV-2 viral replication, because you can actually block the stress response using ISRIB. And you can see that here with ISRIB. With halofuginone, it has no effect on the downregulation of the percent infection, showing that the stress response is independent of this. So halofuginone specifically reduces the heparan sulfate biosynthesis. It's actually already available, and it's been evaluated for phase 1, as I mentioned. What they're now doing is partnering with a biotech firm to, and I'm not involved in this, so I don't even know what even the progress of this at the moment, but they're making steps towards setting up clinical trials as a candidate for the treatment of COVID-19. And if you're interested, this is a recent bioRxiv paper that came out, I think, 2 weeks ago or a week. So as I mentioned in the summary of halofuginone within SARS-CoV-2, it has anti-inflammatory properties. It downregulates heparan sulfate, but it also inhibits the viral replication of SARS-CoV-2, because when you add the proline tRNA to the culture media, and you essentially can rescue the downregulation of the SARS-CoV-2 binding. This is the graph here. So knowing that that's the mechanism in that system, the question is, how does halofuginone kill myeloma cells? So we also see a rapid induction of the amino acid starvation response, similar to the Th17 paper that I showed previously. So this is treatment of AMO multiple myeloma cell lines, and then bulk RNA-seq is performed. So it's a very fast-acting compound, and it works at sub-nanomole doses, and we see death within 24 hours. So you can look at the transcriptional response. You won't be able to see any of the genes. I just want you to focus on the magnitude of the response and the numbers of genes that are differentially regulated. There is an increase in the magnitude at 24, but actually there's still a substantial response after 6 hours. And we can quite clearly see that halofreginone is inducing this stress response by the upregulation of a number of amino acid and UPR pathway genes. And this is definitely ATF4 driven because you can overexpress it and you see the induction of the stress response. What we're looking at now, and I don't have the data yet, is looking at the shift in metabolic function within the myeloma cells, because you should expect to see a change, at least in the TCA cycle, and the cells moving into more of an apoptotic and anergic state. So that's hopefully an introduction to why we are, well, a background to why I'm working on single cell sequencing, which will be apparent in a second, because that's all in cell line models. Really, cell lines are great for formulating hypotheses, but the problem is multiple myeloma is so much more complex than that. It actually has the interaction between the immune system and the myeloma, which then there's plenty of studies shown that this actually helps the myeloma escape or develop drug resistance. So we really want to look at this and look specifically at the effect of the compounds in a more ex vivo setting. And the problem with myeloma is when you look at the bone marrow, there are so many different cell types within there. It's very difficult to sort all of those out using flow cytometry and then treat the cells separately. So that's our rationale for wanting to use single cell sequencing, because it allows us to really understand the heterogeneous tissues. We can identify very rare cell types, specifically, hopefully, the clones within myeloma. And we can then look at the cellular composition changes that occur with and without treatment. In addition, it allows us to also look at something across a temporal process. So we are taking patients before drug resistance, after drug resistance, and then tracking those patients over a number of years to really look and identify, what is the mechanism of drug resistance in certain treatments. So the single cell approach that we're taking is to use a droplet-based system. So this was initially published by Klein et al. And McCusker et al. In back-to-back 2015 Cell paper. The principle is very simple. So you have a stream of lysis buffer, which contains beads, and these beads have an oligonucleotide that can capture mRNA from a cell. You then have a stream of cells, and then a stream of oil. And the flow rates of the oil, the cells, and the lysis buffer are such that you can actually get one droplet, which would have either a cell or a bead, or a cell and a bead. And when there's a bead and a cell, you will then capture the mRNA when it's lysed within this droplet. And depending on which system you use, for 10x, the droplet becomes the reaction vessel, whereas in the Klein et al. And the Costco, everything is done on the droplet outside of the bead. And the beads are very simple in construction. So you have a poly-A site. So the beads are synthesized in 5' to 3' direction, which is actually not the way you would normally synthesize an oligo. Hence, it's slightly more, it's more expensive to produce these oligos on the beads. You have a poly-A capture site, which is just a load of T's. Then you have a PCR handle, which will then allow you to PCR amplify any of the captured mRNA. You have a barcode sequence, and this barcode sequence is specific to each individual bead. So you can then computationally work out which bead which cell and which read belong to which cell. Sorry, garbled that slightly. Then you have a unique molecular identifier. And this UMI is there to remove PCR duplication events, because if you see the occurrence of, and this UMI is specific to each individual captured mRNA. So if you see, say, 2 UMIs that are the same, you know that's been amplified 3 times. So you can collapse that into 1 count rather than 3 counts. We invested within the botnet into single cell sequencing systems. We have 2, well, we have a few more as well, but the main 2 that we have is 10x, which I think is probably the market leader at the moment. That's the gold standard. The problem with this is a closed system. So you just buy the reagents that 10x sells. You can't change any of the underlying technology or methodology. We have also bought a Dolomite Bio Nadia system. This is a commercialization of the dropseq method. So it's a more open system. And this allows you to play around with the single cell sequencing. And this is what we've done, and I'll mention at the last 10 minutes of the talk what I've been doing here. So the experimental design is actually quite simple, that we've chosen to investigate halofugenome. So we take fresh bone marrow sample from the lab. It's then sent to us. It's processed by Martin Philpott, who I collaborate with quite closely. The cells are then cultured for 24 hours in DMSO. So this is the full bone marrow, and then treated with DMSO and halofugenome for 24 and also for 48 hours. We then load 20,000 cells into the 10x system for transcriptomic analysis. This should give you around about 10,000 expected recovery. And Anna, a student in the group, has analyzed the majority of this data using the single cell sequencing workflows that we have. If you're interested, you could look at them here. They're all on GitHub. But the main way we process our data is we don't use Cell Ranger or any 10x approaches, because I much prefer using the pseudo alignment methods, using Callisto bus tools or Salmon Alevin. And then one of the most important things that you need to do during single cell analysis is get the quality metrics right, because any bad cells will completely confound and mess up your downstream analysis. So we typically spend around about 50 to 70% of our time focused just on QC to make sure that all of our metrics are correct. And these vary depending upon which different types of experiment you use, typically on which model you're using. The downstream analysis, I'm not, for this particular work, it's nothing, to be honest, novel that other people in the single cell field aren't using. I have my own custom QC scripts, but apart from that, I'm either using ScanPy or the CRAT workflow in R. And then really trying to, the biggest problem in single cell isn't analyzing the data. It's actually ascribing biological information to the data. That's a really difficult aspect of the work, and I'll touch on that in a few slides. So the first visual plot that people like to see when they're looking at single cell sequencing is either a UMAP or a t-SNE. And all these plots are doing is taking that really high dimensional counts data and representing it in a 2-dimensional space. What we then do is we then cluster, and then what you should be able to see is nicely defined clusters coming out of both your UMAP and your t-SNE. And this is exactly what we see. It's very highly granular data that we have. So what we now need to do is try and ascribe an annotation. What actually is this cell type 0? What is 2 versus 5, et cetera, et cetera. And this is the challenging aspect. This is where you typically get a PhD student and put them in a room for a week or so and then say, please tell me what the annotations are. It can be quite laborious, but once you've done it and you've assigned an annotation, that's when the magic can start to happen. You can start to really do lots of differential analysis, composition analysis. But to help us with this annotation, we actually use a number of published studies where we have used classifiers. For instance, here I'm using Clusterify R, where we can then use data that's already in the public. For instance, I think in this, it was PBMCs. They don't have data sets that align with bone marrow, but the closest one is PBMCs. And so using this particular approach, it's spat out that this could be NK cells, This could be erythroblasts, this could be B cells, this is unassigned, this is monocytes, this is T cells. Not terribly informative, but at least it does give you an understanding and a start of where you can move. And you can apply different annotation packages. So for instance, single R and SC classify. They are then giving a little bit more granularity, but you can see that, you know, this is identifying them as cytolytic T cells, CD4 T cells. Here, this is identified as an NK and just T cells. So there are differences. But what it allows you to do is to manually now go through each individual cluster and then ascribe an annotation based on, unfortunately, it's a lot of reading. It's a lot of interacting with my colleagues who maybe are more experts in particular monocyte subsets or T cells. And it takes time. And it should be done very robustly, because if you get any of this wrong, it then ultimately does have an impact on your downstream analysis. So for instance, for this particular project, we're very interested in defining where are the myeloma cells. There aren't any data sets that we can go to that have myeloma cells defined in them. So we have to definitely do this by ourselves. And so we typically just look at panels of markers and then say, okay, using a number of different markers, where do we think the myeloma cells are? Here's 5 markers here, and they're all highlighting this population down here as being highly expressed. We then look at a few more, and then we start to build a picture saying, well, actually, this is potentially our myeloma population. We'll then look at this in minute detail at the cluster level, not just on the global population level, and then start to then add in better annotations to what we see. At the moment, this is still ongoing work, but you can see, you know, we've now identified cluster 5 is definitely the multiple myeloma population. Cluster 11 is more plasma cell-like. And then what we can do is then, once we have all of these different subsets annotated, we can then build a classifier. And this classifier will help us classify any new data coming in. So we're now ramping up the single cell sequencing analysis work that we're doing. And we can use this classifier to help us in the future. So now you can then, okay, once we've identified where the myeloma cell population is, we can then start to look at this in more of a biological context. Okay, so for instance, the halofugenone, we know that it induces an ATF response. Let's look at the ATF genes. And so you can see after 24 hours, we get a significant increase in ATF 3-4 activation, indicative of this amino acid starvation response. And again, it seems to be very restrictive to just the myeloma population. We can also look at composition. So not just, okay, what is the expression of the amino acid starvation response, but actually, do we see reductions in the myeloma cell population? And we do. So cluster 5 is the myeloma cell population, and you see with halofugenone treatment for 24 and 48, there's a definitive reduction in that population. So the biological implications of this are that actually halofugenone acts preferentially or has a preferential effect on the myeloma population. And largely, we do see a transcriptional response within other cells, but it doesn't induce cell death, which is hopefully positive. So I think hopefully what I've shown is that we can now define multiple myeloma populations appropriately. We have the ability with single cell sequencing to investigate both the immune compartment as well as the myeloma population. The great thing about this is that you can actually then look at, you know, drug responses. You can look at this across a longitudinal time point and really understand the disease in more detail. However, there's a big caveat to short read single cell sequencing that I've just shown you. And that's that we just really can't, we don't have enough information that will allow us to identify clones. because all we're doing with short read sequencing is essentially 3' or 5' tag counting. You're just looking at the ends, the extremities of the genes, and then counting, okay, that is that gene X, gene Y, et cetera, et cetera. We don't have any further information that will allow us to say, actually, there's a translocation here. We only see this within this particular subclone, or there's a mutation within here, and that we see this within a clone. So what I am going to talk about in the last, well, hopefully 5-10 minutes, is some of the work that we've just recently submitted to bioRxiv and is in revision at the moment, looking at the long read single cell sequencing. So long read single cell sequencing is, at the bulk level, is revolutionizing analysis, because you can, at the RNA level, you can perform true mRNA, full-length mRNA isoform resolution, which you can infer from short read to bulk, but it's a lot more difficult. However, there's a big problem with Nanopore, and that's its high error rate. Unfortunately, the barcode and the UMI, you need around about a 98.5 and above accuracy in the sequencing base calling for this to be useful for single cell sequencing. However, this hasn't stopped groups, and actually I recommend you reading this paper. It's a really nice implementation of how they have used computational approaches where they first sequence with Illumina and then sequence with Nanopore, and then use the Illumina barcodes and UMIs as a guide to error correct the Nanopore ones. The problem with this is that you need to sequence 2 different platforms. That requires you to make 2 different libraries, and it adds to the complexity of the workload that you're doing. In addition to this, defining the barcode's fine, but the UMI is a little bit more tricky because it's actually generated at random. And so it's not always easy to define the UMI accurately within the Nanopore read. So this is where I came along, looked at it, and I thought, well, why don't we not try and find where the sequencing error would be within the barcode and UMI without using Illumina, and then use that to correct the sequencing. So we build up the PCR handle with single nucleosides, and then the barcode and UMI is built up with blocks of nucleotides. We've shown within a recent paper that, by archive paper, that you can build this up very effectively with blocks of dinucleotides. And then the poly-T region is just built up with single nucleotides. Why this is important is because it allows you to identify very nicely the errors within either the barcode or the UMI. So you can see here, this is the original barcode in UMI. And just by, this is a hypothetical sequenced read, AA, it matches perfectly. You've got base pair complementarity, TT, but here you've got an AT. So if you didn't know the top sequence, you would say, well, actually, AT, there's got to be a sequencing error there. And then you compare it to the original and you can see, it's got to be an A. And you can do that for both barcode and UMI. The barcode correction is essentially a similar approach to how the Waldman group do. I do a 2-pass analysis strategy where you identify all the barcodes with errors and all the barcodes without errors, and you bin them into 2. You then do a second pass where you use the barcodes without errors as a guide to error correct the barcodes with errors. And then you can then collapse that barcode down into single bases. UMIs, I'm using an adapted directional network-based method that was first proposed by UMI Tools in the Smith et al. 2017 paper to correct the UMI duplication. So it adds an extra UMI into the network. So then you can see here, you would have the original UMI CCGG, the sequence molecule, see if there's an error here, CCGA, that would be split into 2. So you're actually adding an extra UMI here, CAT, to the UMI network-based method. So this is a representation, a graphical representation of 2 UMIs, one without sequencing errors and one with. And you can see that when they differ, you can create this network and then collapse all of these UMIs into the one UMI and this into a separate UMI. The circles represent a theoretical frequency of seeing that UMI within your data. We can show that this method works using simulated data. So the original directional method, where it's just accounting for single bases, doesn't do a great, isn't great when you then use, when you have a sequencing error of above 8%, 10%. But the directional paired method is very robustly accurate. And this is simulated corrected versus the ground truth. The gold standard of assessing whether the experiment works is actually do an experiment where we do a mixed species experiment where we encapsulate human and mouse cells, and then we look at the ability of the method to correctly assign each barcode to either a mouse or a human. And if this wasn't working effectively, you would see that we would get a huge increase in the numbers of mixed species. So the method wouldn't be able to correctly assign to a mouse or a human UMI, which you can see here on a barnyard plot. All the cells that have a human read are on the bottom, and all the mouse cells, each point is a cell, is on the left axis. Why is this important, the single cell? Well, actually, it allows us to now start looking at transcript usage, which you cannot do with short read single cell sequencing. So what I've done here, I mixed 3 myeloma cell lines, and I'm using this just to see if I can resolve those 3 different distinct myeloma cells. And we can see quite clearly that we can do this both at the gene expression and also the transcript level. We can also identify for this HLA-DR, which is a target for myeloma, where which isoform is being expressed and within which population. And that is quite clearly within this JGN3 cell. And the most common isoform is this 3334. However, it actually allows you to do a lot more. So it allows you to capture more information per cell. So we've shown that we can also look at fusion transcripts. And we've demonstrated this in a proof of principle using Ewing sarcoma, where we can detect the major EWS FLI fusion transcript. So this drives the development of Ewing sarcoma. We can see this in the presence of around about 20% of our cells, and we can even detect which actual fusion event it is within this cell line. And so it's very well characterized cell line, and it phenocopies what we would be expecting. I'm also looking at this in the context of IgG, TCR, and BCR rearrangements within myeloma. So because we have the full length sequence, we can reconstruct the TCR, BCRs. And I'm just showing that different cell lines actually have a different pattern of IgK and IgL translocations. So the benefits of single cell sequencing, hopefully I've demonstrated that, you know, it opens up the possibilities of doing new biological applications. I'm also working with NASA in the WIM, who works on variant calling methods, to start variant calling using our exomes that the RNA-seq reads, the full length. And we think we can detect the presence of clones that are developing over a drug resistance process. So the caveat to this at the moment is that at 1,200 cells, sequencing on the Promethean versus Nanopore versus Illumina is actually cost comparable. But when you get above 1,200 cells, the sequencing costs then become cheaper for Illumina, but then balloon for Nanopore. However, hopefully, there's been so much progress on the Nanopore side that they'll hopefully produce bigger and larger machines soon. So what's next? So as I've said, I'm investigating this with Celgene BMS, looking at clonal resistance in drug populations. One of the things I'm also developing is a dual capture system where we can capture the long read RNA at the same time as also attack, cut and tag at the single cell level. And we're establishing a spin-off company to commercialize this. So myself, Martin, and Udo are founders for Cerealius Genomics. We're developing this company we've had some funding from Innovate UK. We're now going through the venture capital route, and this is in a very good collaboration with ATD Bio and Professor Tom Brown in chemistry. And that's essentially it for the talk for the moment. I hope a little bit overrun, but I'd like to acknowledge Uda Opperman specifically, Martin Philpott, a number of collaborators, particularly the clinical collaborators, such as Karthik and Sarah Gooding. And again, I've been incredibly lucky to have had some fantastic collaborators and some really good, some funding to pursue some of this work through various different charities, but also pharma companies as well. And the Medical Research Council that funds me at the moment. Cool.